Today we will be presenting about the Influenza A. The team members will be me, Julie, Punch, and Jam. We will all talk about the 5 main topics. Here come the first topic, the cause of the disease.
Influenza A disease caused by Influenza A virus. Influenza A virus are member of the family of the mixed oxidative. This family represents envelope viruses that consist of 8 segment negative and single-strand RNA. These capsids are encircled by the M1 matrix proteins and by a host-derived lipid bilayer envelope in which the viruses surface glycoprotein, hemagglutinin, and neuraminidase as well as M2 matrix protein are embedded. Viruses are not cells and cannot replicate without help, but they have nucleic acid and can envelope, so it neither prokaryote and eukaryote cell. As you know, there are 18 HA subtype and 11 NA subtype, so many different combinations of HA and NA protein are possible. The virus have wide host range such as bird, human, horse, and dog, but there are only H1N1 and H3N2 are currently in general calculation among people. Life cycle of IAV like other virus. First step, the IAV enter the host cell by blending with surface receptor processing icyclic acid moiety. Then viruses are internalized by endocytosis and uncoating of the virus. And the vRNA was released in cytoplasm and imported to the nucleus, thus synthesis of the RNA in nucleus. After that, the viral mRNA is translated in cytoplasm and cellular ribosome synthesis nucleoprotein.
So the next topic will be history and current research of the disease. Who discovered the pathogen and disease? Although the people know the influenza symptom more than 1,000 years, the first human influenza virus was isolated by Wilson Smith, Christopher Andrews, and Patrick Ledrow in 1933 because they found a suitable host for propagation. How long have we known about the disease? So it was from 1918 the influenza H1N1 was emerged to today are 102 years. Now I will talk about history of influenza A in the world. The H1N1 was first identified in US in 1918 and spread worldwide. 500 million people was infected, and 17 million deaths or 3.4% mortality. In 1957, a new influenza A H2N2 virus emerged in East Asia. The virus was comprised of 3 different genes from H2N2 in avian. The estimated number of infected people was 478 million with deaths, 1.1 million worldwide or 0.23% mortality rate. The 1968 pandemic was caused by an H3N2 virus comprised of 2 genes from an avian virus. 200 million people was infected. The estimated number of deaths was 1 million worldwide and 0.5% mortality rate. In the spring of 2009, a novel influenza A H1N1 virus emerged. It was detected first in the United States and spread across the world. 500,000 people was infected. A mass vaccination campaign was introduced in addition to the prevalent seasonal flu vaccine by November 2009. Today, only 2 influenza A virus subtypes H1N1 and H3N2 are currently in general circulation among people. Next, how has our understanding of the disease developed over time? A large number of studies have revealed that the evolution of influenza A virus is mainly mediated through the mutation of virus itself and the reassortment of viral genomes derived from various strains. The highly pathogenic influenza virus has resulted in stupendous economic losses due to its morbidity and mortality, both in humans and animals. Influenza viruses fall into a category of viruses that can cause zoonotic infection with stable adaptation to humans, leading to sustained horizontal transmission. The evolution of influenza virus depends on 2 mechanisms, antigenic shift and antigenic drift. There are 2 types of mutation of influenza virus. The first one is antigenic drift or small changes. It means the change in the genes lead to changes in the surface proteins of the virus such as hemagglutinin and neuraminidase and produce viruses that are closely related to one another. Antigenic drift is the main reason why people can get the flu more than one time, and it's also a primary reason why the flu vaccine composition must be reviewed and updated each time. Antigenic shift is major change, resulting in new proteins and can result in a new influenza A subtype in humans. One way shift can happen is when an influenza virus from an animal population gains the ability to infect humans. Such H1N1 virus with genes from avian, human, and swine emerged to infect people, causing a H1N1 PDM. So type A viruses undergo both antigenic drift and shift are the only influenza viruses known to cause pandemic. Pictures show the evolution and relationship of human influenza with influenza in the other animal. Solid red arrows indicate the evolutionary path of human influenza A virus lineages. All the pandemic viruses since 1918 contain gene segments derived from the 1918 H1N1 virus. So this graph shows the number of cases positive with influenza virus. Number of influenza A positive was shown in blue color graph. It's mean more than 20,000 sample positive in 2019 to 2020.
The first case of influenza 2009 was found in United States and it quickly spread globally. As you can see in the picture, the influenza subtype A was found in many parts of the world, such as United States, Africa, and Asia, including Thailand. From the CDC information, the mortality of influenza A virus from 2009 to 2018 was 0.001%. Morbidity rate was 0.01% and not found new case. From the information of Rajatevi Hospital, data were collected about history of fever at outpatient department of Rajatevi Hospital between June and August 2009. The present study was divided into 3 phases, as you can see in the pictures. Influenza virus may be transmitted among humans in 3 ways. 1. By direct contact. It means transmission by infect droplet. Droplet can come from sneezing, coughing, or talking. The infect droplet have ability to infect others as far as 6 feet away. 2. By indirect contact or contact with contaminated objects such as doorknob. Transmission can occur when influenza droplets surface on objects and people come in contact with them. All age groups can be infected, but there are groups that are more at risk than others, such as a pregnant woman, children under 5 years, the elder more than 65 years, the patient who have chronic medical condition, and the immunosuppressive condition. Influenza occurs all over the world with an annual global attack rate estimated at 5 to 10% in adults and 20 to 30% in temperature region. Influenza is a seasonal disease occurring tropical winter months. It affects the northern hemisphere from November to April and the southern hemisphere from April to September. In tropical region, there is no clear seasonal pattern and influenza calculating is year-round. Typically, there are several peaks during rainy season. Influenza A is a acquired communicable disease.
For the pathology, influenza A infected the cell in upper and lower respiratory tract, such as epithelium cell of trachea and bronchi, alveolar epithelium, goblet cell, alveolar macrophage, and pneumocystis type 2. About the damaging, this virus replication at the epithelium of trachea, bronchi, and pulmonary alveoli cause clinical signs and symptoms of pharyngitis and tracheobronchitis. Then it infects alveolar epithelium cells, cause capillary thrombosis, necrosis of alveolar wall, and development of hyaline membrane, cause necrosis of alveolar epithelium, and cause respiratory failure. The time sequence of this disease. The incubation period of influenza A range from 1 to 4 days. These symptoms usually last for 3 to 5 days, although the cough, malaise, and fatigue can last several weeks. Adults can chase the virus for 10 days, but the children can chase the virus for longer than 10 days. The major signs and the symptoms. The clinical presentation of influenza virus infection can be various according to the patient's age. From the study, major sign of influenza A in adult is repeat onset of fever more than 38.5 Celsius, 96% of patients, severe non-productive cough, 86% of patients, severe headache, 66% of patients, malaise, 78% of patients, diarrhea or vomit, 28% of patients. Other clinical signs such as sore throat and rhinorrhea are included. Lastly, we're going to talk about response and treatment. Immune system against IAV. Immune response to IAV divided into two parts. The first line of defense against IAV infection is innate immune system that comprise the physical barrier, phagocytic cells, cytokines, interferons, and IFN simulated gene. The second line is adaptive immunity, which mediated by B cell and T cell, characterized with antigen-specific memory cell. For the treatment of this disease, is conventional medicine by using antiviral drug. Antiviral drug can divide in three major groups: inosine 5 monophosphate dehydrogenase inhibitors such as ribavirin. Second is M2 protein inhibitor such as amantadine. And last one is neuraminidase enzyme inhibitor such as oseltamivir and peramivir. Antiviral drug must given within 48 hours after get sick, but given after 48 hours still health. The drug can reduce hospitalization and mortality rate of illness case.